I'm going to record. Okay, I try to record like the first part of the lab where like I'm telling you guys about what we're doing today. I haven't done that in the past. I don't know why nobody has done it in the past, because this way, even if like, so if you're gone and you didn't get the full rundown of the thing, it's available to you. But also when you're going back to study, if that's part of what helps you learn the thing with me talking about it, then it's available to you. If you don't need it, you don't need it. We'll try it out and see if it's helpful for people. I don't know. And I'm posting pictures of it online as well. You're welcome to take pictures too. It's not like proprietary information. Technically it is, like, don't share it online. You guys know how this stuff works. Yeah, it belongs to the university technically, but you're welcome to take a picture and use it for your own. Yeah, so let me close this before I forget. We're going to talk about what we're going to do today. What we're going to be doing today, we're just not dealing with what we missed. We're going to pretend like that didn't happen. Basically, that was never a thing. I don't know what is going on. Here it goes. Save. Okay. What they did, what my Monday, Wednesday people got to do that you guys don't do. We took cotton swab like our feet or our phone or whatever it was, and swabbed out like these plates to grow to see what was going to grow. Okay. And then they got to look at it today and apply colony morphology descriptions to it. I didn't have anything to turn in or anything. It's just for you guys to like, oh, okay, yes, this is that, or that is this. Now I see and I understand. That's what it's for. So I will let you guys, these are some good ones. I'm going to put them up here if you guys want to see, okay, if you don't. But that's there for you guys to look at and apply that colony morphology stuff. What is colony morphology? Something that we can use to describe the colonies. They're different for a lot of different species based on whatever species we're talking about for bacteria. So that's something we can use to identify a bacteria, part of the thing we might use. So how do we describe that? By their shape, we might talk about them being round or irregular or punctiform. Punctiform means like little tiny dots. Margin, that's the edge, whether it's entire or smooth, same thing there. Lobate, literally like has this like lobe like appearance as it goes around. Filamentous, long filaments coming off, and rhizoid is like filamentous but branching off. At surface, the surface of the colony, is it smooth, rough, or wrinkled? The texture of the colony, if you were to poke at it. Moist, mucoid, got that stickiness to it. Buterous means like butter. Dry, shiny, dough, all of this can apply to it as well. Elevation, how raised it is. So if it's totally flat on it, if it's raised up, like if it's totally flat onto the auger, it's raised up onto the auger. If it's convex, if it is pulvinate, which means very convex, like super piled up. Umbinate, which is shaped like a fried egg, so flat on the edges and convex in the middle. And then crateriform, which is crater, which is like, looks like a multi. Rises up and then it has sticks. There's some on there that look like that. Obviously some of these will have like more than one. Like you could have something that was I don't know, raised, and you might describe it also as convex. Moving on, color and translucency. Those are pretty strange. Okay, so if you guys want to, you can go look at those plates and try to see how you describe some of those colonies and apply that to those colonies over there. You will have your unknown projects in the second half of the semester, where I'm going to give you guys a mixture of a gram-positive and a gram-negative bacteria mixed together in one tube. And you will have to separate them out and then identify them. And part of that is going to involve writing about their colony morphology. And based on what you write, if you just write gram and then submit that to me, I'm like, you want full credit for this? Like, really? After I just went through all that for you? No. So you get the idea with that. So that comes up a little bit later. Okay. Aseptic technique. This is preventing contamination, essentially. That's all that's for. The whole thing here is trying to keep things as sterile as we can. We have in our drawers, let me just find it. We have in our drawers a needle on, it's like an aluminum stick with a needle at the end, and then an aluminum stick with a loop at the end. So everybody should have one of each. Today we're going to be using the loop. So I'm just going to stick with this one for now. We don't use the needles for a while. Okay, so you flame your loop to glowing orange. And you want it to be glowing orange the whole way down the whole wire, the whole thing. Okay? Stick it through, get it glowing orange, let it cool for about 15 seconds. If you put it into the liquid while it's still hot, you'll hear it. If you hear that, you did a bad thing. That can cause the bacteria to aerosolize when you do that. Also, it kills bacteria, obviously. And so be careful with that because you want to avoid it in general. Yes, there will still be bacteria in there for you to use. If it happens to you, just be like, whoops, and continue on with your life. Like, it'll be okay, but just try not to keep doing it. Right. Let it cool, and then you will open up your tube. You'll flame the opening of your tube. That's to keep that whole area kind of as sterile as we can. Pass it through the flame like 2 or 3 times. And then you'll put your sterile loop down into your sterilized opening of your tube, down in, try not to touch the sides too much, try to touch them at all, and then go down into the liquid to wash up, swish up, whatever it means to you, a little bit of liquid onto the end of the loop. Now, how do you get liquid onto a loop? I mean, it's going to look like a bubble, like you were going to blow bubbles if you look at it. So that's what it'll look like. You get it up like that, and it pops. That's okay. You still got enough bacteria on there, I promise. You had enough, like we verified there was liquid on there, there will be enough cells on there for you to do the thing. You don't have to go back in and like fret about it or anything. It's fine. Anyway, so you will get that liquid on there and then you flame your tube again, keep it sterile, and then put the lid back on, put it down, you don't need it anymore. And then we will go and put this wherever we're putting it. Today we're putting it on plates. Okay. We will be streaking our plates with the loops. So you will open up your plate. Don't worry, I'll show you all of this in actual like I'll do this all for you to show you. But for now, words. We'll streak it on the plate and then you will flame it again. You're done. Okay, sterile, you can put it away. If you are not totally done and you want to go on to do another thing, or even redo that same tube you just did, still need to reflame it in between the tubes. Okay, reflame anytime you get, and that's what I have written down here. Flame the loop between each get. Anytime you go in to get a sample, you should have flamed it. Okay. Keep it sterile, keep it sterile. Cool. So if we're putting that into a broth, so that's how we would do the auger. We would gently touch it down onto the auger at an angle with the like wide side, like facing flat onto the auger, and just wiggle like spirit, wiggle back and forth. Okay. That's how you put it down an auger. If you're using a slant, which is like what I have over here, and we'll be using these later on in the semester. If you have a slant, these are solid auger as well. You can see here, like it's not liquid and it's slanted. So on those, you would just take your sample and go to the bottom of the slant and come up. That's the best way to do that. And we'll review that when we do those again. If you are, let's try, and if you're putting it into another liquid, you just switch it around. Right, that's sterile. Be sure you flame your tubes when you open them. Yeah. So if you were, however, we're getting our stuff from tubes, liquid, that's fine. But at some point, you're going to be getting from a colony or growth on the plant. When that happens, you're getting from a solid surface. You just want to touch on to the colony or the growth that's on your slant, just enough to pick up enough that you can see that there is some on there, but just barely. You don't need more than that. Some people try to pick up like an entire freaking colony or like just really scrape down that whole plant, and that doesn't work for me because I have to grow more for you later, and that's a huge pain. So, so yeah, you only need to touch it a little bit, and you might want to have enough for it to do whatever you're doing. Yeah. Other than that, you would take that solid base colony and do the same stuff with it into the broth or onto the plate. So that is the idea of a septic technique. We'll talk about what we're going to do with it here in a moment. The other part that I want to talk about is the street plate. We said on the question, right, that it's for isolation. So how do we do that? What does that mean? I'm going to go ahead and show you guys how the street part works. Got your plate and it's completely bare. We'll onto the auger surface. We've flamed our loop. We've gotten our sample. We've got our liquid on here, right? We're going to just, our first thing, just lay it down, however. Get it on there. That little section there. The idea is just get a bunch of cells down. Okay. Then you're going to flame your loop. Right. Your instinct is going to be to put it back into a tube. That is not what we do. Here, when we're doing this, for isolation, we're trying to spread out so that we can get like individual cells that can grow into individual colonies. That we can later pick up and do our studies. Okay, so got that first one. We flamed our loop. Do not put it back in a tube. The second thing, you go into that with the flame sterile loop, maybe once, maybe twice tops, and then just squiggle it out like 3 or 4 times. All you need for the second. Flame it again. Back into this, not tube. Straight on here, go in. Once or twice, squiggle it out. And that's your number 3. Flame it again. And then 4 is going to go in once, maybe twice, and then squiggle out into whatever remaining space there is. Most of your single colonies will appear in that 4 area, that 4th one. Okay. We call this a, I'm saying 4 quadrant. Call this whole thing a quad plate or a quad streak. Quad streak. There's a streak plate, is done with the quad streak method. Gone through it, like lay down this one, pull through 2, pull through 3, and then 4 and pull through and squiggle out. How I would describe it, I don't know. That is for isolation. Okay. That is a technique that we've learned also. We've learned a aseptic technique. Now we've learned how to do a streak plate. You will be doing the streak plate when you guys get your unknowns and you want to separate them out, gram positive and gram negative. You'll be doing the streak plate to get the full column work for us. And yeah, you guys are going to be doing that all by yourselves. I mean, I'll be helping you, but I'm not going to do it for you. All right. Aseptic technique. These are your Bunsen burners. I'm sure you guys are familiar with what a Bunsen burner is. Most of you have taken some, you're supposed to have taken chemistry. I'm pretty sure you use Bunsen burners there. Our gas here, our lab is set up a little bit differently than the other lab. Obviously, this is a way newer lab. So our gas is here. If it's facing out that way, it's off. If it's facing in this way, it's off. All the way it's on, face it down the actual line of the Bunsen burner. Yeah, I saw that. One of the students, I turned it down way low. And you can adjust the height of your flame with the little wheel at the bottom if you need to. Okay. This is your striker. This is how you're going to light your Bunsen burner. You're going to feel like idiots. I'm just warning you. Like it's going to be, it is this way. Every time that I come in, I'm like, okay, we're going to do this and that. Once you guys get comfortable in here, you're doing stuff kind of basically on your own. Like, okay, do this and that and the other. And you're going to be like, click, 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 click. And it's going to be going for like 10 minutes straight. Like it's going to be ridiculous. So here is my advice. When you use your striker, mine is a good one. So if you absolutely really need to use mine, you can, but I don't want it to get lost or whatever. I do a lot of filtering, so I like to have mine where I know where it is. But you will hold it with 2 hands, trust me. Okay? I know you can do it with 1, but just trust me, hold it with 2. Because then you can use most of your force from your other hand on the one that is going across there, you see. Like if I hold it loose like this, I don't get quite as many sparks as if I hold it like this and bring it towards you like that, right? So that's what you want is the sparks. So push the one that's going across in towards the thing while you're doing it. I know it sounds like, of course you would do that, but you'll see. Okay, when you light it, also don't hold it like this. This is just causing the gas to not be able to flow how it wants to. Hold to the side. Close. Okay. Notice it took me 2 clicks. You are not going to take 2 clicks. So that's okay. That's okay. We'll get better at it. So now we've got our Bunsen burner lit and we can start working on whatever we're working on. I'll pick up my plate. And I will get this heating up. Now, one thing I want you to notice. One thing that you will notice is that if you have it high up, like above, the flame has like a little cone in the middle of it, right? Where there's not like a bright flame. There is no flame there. That's why. So because it's coming up around that circle, not to the middle of it. So if you hold your thing like this,
you're not gonna get orange forming all the way across. It's just gonna form on the edges of where the flame is. So you have space. Be aware of that when you were doing this. It's better to come up higher in flame so that you can get better coverage of your wire. And these ones I have are thicker wire loop. These ones take forever to keep. The thinner wire loops are quicker, but the problem with them is they break here. Just there's pros and cons, whatever. And you got that done. Let it cool for 15 seconds or whatever. Then you're going to grab your tube. I recommend flicking it a little bit at the bottom to mix it up a little bit because the bacteria settle on the bottom. It's a better result that way. Then what you're going to want to do, hold your loop like a pencil, sort of. I mean, that's how I hold a pencil. And that's fine like that. And you'll grab your tube. When you pull, number one, first of all, these lids are not like on securely. Some of these tubes, when you lift them up, the lids will just come off. So be aware of that. It's not secure. If you tip your tube too much, it'll just pour out, even with the lid on. So don't tip it too far. And be aware of that. People seem to do it. I don't know how. And then always, when you can lift it up a bit by that tap, but then grab the glass before you move away from your rack. Okay, just be safe. It's secure. The tube will just fall out. Okay, so I like to push up the tube a little bit with my fingers as well as on my left hand, as well as grab with my pinky of my right hand. This allows me to hold onto the lid. Don't put your lids down on the table. You're going to risk contaminating. This might have, staff on it. And now I've got it all over the table. Or somebody else had their tube down earlier, and now it's full up on this, and I'm putting it in the contaminated stick. So hold on to your lid while you're doing it. Then you will take your tube and pass it through the flame about 2 or 3 times, back and forth. And then you will hold it at a little bit of an angle. It's just easier to get in there that way. Go down in the fluid, and I just kind of swirl it a little bit. Try not to touch the side. Say if you do. I'm just trying not to. And flame it. Back and forth, real gentle. And then I close it with my thumb. Now that's done. Now you don't have to worry about that tube. It's taken care of. Okay. Your auger, always auger up. So essentially, the whole thing upside down. Always. That is because the lid collects condensation on them whenever we move them from like the refrigerator to room temperature. And that condensation can contaminate or cause contamination by dripping all over the thing, spreading everything around. Okay. So that's the idea with that. So always auger up. That's also the surface that you will write on with your name and whatever else you might need to put on any identifiers, whatever. So that auger side. Okay. Then you have it down, lid down. You'll pick it up, and then you can access your actual auger surface. Okay. We've got our liquid on here. I picked up number 18. So on my 18 area, which you guys are going to do here in a minute, all you do is gently touch it down onto the auger and just squiggle it a little bit. It does not have to look pretty. You just have to get it down. If you don't want to do a squiggle and you just want to do like a line, I don't care, as long as you get your stuff down. Okay. Now we've done that area, we will put this down to keep it clean. You don't want to just leave it facing up. And then you will flame this to get it sterile. If you guys are done after this, this was your last tube of the day, then you would want to, you don't have to wait for this to cool to put it in your drawer. Okay. Nothing is going to get killed in that plastic bin in your drawer. Some people at the end of the day have a tendency to just leave their thing here, and at the end of the day, I have to put away your stuff. I just don't want to. So you will just put it back in the little tray in your drawer. Okay. Otherwise, you just flamed it and you're ready to go to the next thing. Then it's sterile and you can move on to the same thing the next one. Okay. That's how aseptic technique will work. I will leave that on. Right, so what are we doing with that today, right? What we're doing today is we've got a whole shitload of plates here, and we're going to essentially, like it says up there, stack of 6, see at the very top on the right, stack of 6. You're going to label them just like I had that plate labeled on your auger side, make a cross,
18, 19, 27, 29. On all six, each of the six, each one has that on. And then after you have that on there, you're gonna go through and put the 18 tube on the 18 spot for all of these, and so on and so forth. How I recommend that you do this is label it all first. Then if you have 4 people at your table, one person does all the 18s, one person does all the 19s, whatever. You make sure you just pass the plate around until they're all done. If you guys have less than that, just work out who does what numbers and whatever works for your group. Okay. So you could, like 2 people do 8, like 1 person does 18, 19, whatever. So that's how you get it done. So all 6 of those plates in that stack will have all 4 of those on each one. Okay. These are differential and selective media. I will talk about what each media is and how, like, what the color changes mean and all of that next lab, like when we come to read them and see the results. Okay. I will today walk around with one of the plates from yesterday so you guys can see what they will look like nice and neat, because over the weekend, when they grow over the whole weekend, they kind of get a little overgrown. So I want to be sure you guys can see kind of a little bit what they look like when they're nice and neat. There's one in there that's pretty darn cool. So, but yeah, essentially, you guys know, because I'm sure you watch videos, selective media are media that basically allow some things to grow and not others. So they'll have some sort of additive to them. For example, Staphylococcus bacteria, they do pretty well with salt, whereas other bacteria don't do very well with salt. And then the last 2, so 18 and 19 are gram positive, 27 and 29 are gram negative. So those 2 also happen to be enteric bacteria, so they grow in my gut. So if I tell you that, like, McConkey agar has bile salts added to it, there's bile in it. So obviously, if you would think about it that way, well, bile, okay, well, I think of the gut when I think of bile. So the gut bacteria, 27 and 29, should grow on it, whereas 18 and 19 that grow on your skin probably won't grow in the gut media, right? So that's the idea with those, and that is how it's gonna play out. So that's the selective side. The differential side, that's what lets us tell the difference between things that are growing. They all grow. This one caused a color change, this one caused different color change or no color change. That can be due to if something breaks down a sugar and makes an acid byproduct when it breaks it apart. That presence of acid can change the media from red to yellow. That's a very common color change in media. Okay, that is caused by phenol red. You will need to know that for the final. You don't need to know right now, but phenol red changes from red to yellow in the presence of acid. So that's one possible thing. Yeah, so that's differential and selective. We'll talk about what each of these media are and the color changes and the growth and things like that next. So you guys know how to set that up, and you're going to get confused. Somebody's going to get confused. Everybody does about what to do with it. Each one gets all 4 bacteria. Then we have the other stack. Which somehow you guys have 2. I thought I had 4, but you guys only need 2, so that's fine. The other stack, everybody else should have 4, but they all look like plain, like this. This is TSA, which is tryptic soy agar. It's just a nutrient agar that pretty much anything can grow on. Okay. On this, you're going to do your streak plate. So that quad streak that I showed you on there, you're going to lay it down and then flame it and then spread it out with those next 3, right? You squiggle it out so you get single colony. Single colony will appear in the 4th streak. Yeah, each of you will do one of these. Every person in the lab will do a streak plate. The stack of 6 is for the whole table, 1 for the table. Okay, I think we're ready. I hope that covered everything. Remember to write your identifier on there, whether it's your name or like your table identify, whatever you want to call your table. I don't care. Remember to label everything first. And then
So same thing with these, we'll label them and do one of each tube inside each of them.
yeah. So this is the quadrant thing, like the 18, 19, 17, go on. That's like number 3 up there.
Yeah.
Where I put down like 1 bacterium. squiggle on one, and then I flame it, and then I
Yeah.
kill it, so it's the same bacteria that point through everything. So then you just squiggle it out, try to get single colony, if that makes sense. When I'm telling you guys to do your streak plate, and you guys are probably like, okay, what am I supposed to write bacteria? I'm not confused. I don't care. This is the bacteria that you use. Any bacteria from the tubes. Don't try to like pull it off of your new plate that you just streaked on, from the tubes, okay? And it doesn't matter, you don't have to write down the number either. I just want to see your streak.
Hey. There it is.
Not so bad, not so bad.
Yes.
How do you guys? We don't get